Well, thank you very much, Dan, and thanks for extending the invitation to Marty and I to come in and talk a little bit about some of the work that we both have been doing independently and through some of the work I've been doing investigating nitrate and uranium interactions. Marty and I have begun to develop a collaborative project to begin to look at how here in the state of Nebraska, nitrate may be potentially influencing uranium mobility. And from Marty's perspective, he's mostly interested in just uranium mobility and uranium in drinking water. So just to highlight the nitrate and uranium that's observed in Nebraska groundwater, the data that you see here within the state, this is uranium data that has been compiled primarily from the USGS, so from the NURI database, the National Uranium Resource Exploration Program, as well as the NACWA program. And the nitrate data is a combination of data from many different state agencies here in the state of Nebraska, as well as some of the NACWA data. Nitrate is depicted in blue. Dark blue denotes levels that are going to be above that maximum contamination limit. Uranium, the dark kind of pink color, that's going to denote areas that are above the maximum contamination limit. So this is an interpolation plot of the data that has been measured throughout the state. So it's somewhat predictive and kind of getting a better idea of where nitrate and uranium could be co-occurring, because you see some of the overlap here. So Nebraska potentially could have an issue looking at these correlations between nitrate and uranium. But, you know, to point out, it's not just drinking water. Of course, we could be affected through drinking this nitrate and uranium, as Nebraska does get most of its drinking water from groundwater. But the other thing to consider is that food quality could also be potentially affected by uranium in irrigation water, as some food crop, tomatoes, squash, lettuce, and radishes, has been demonstrated to take up uranium and accumulate it into biomass. So there is an issue not only with drinking water, but potentially food crop. But I want to point out that Nebraska is not unique in the context of uranium found in groundwater. If we take a look at nitrate and uranium throughout the High Plains aquifer, and this is some work that a graduate student in my laboratory conducted, compiling a database consisting of over 256,000 samples from over 77,000 locations. And this is through many different state agencies. Spent a lot of time contacting different states, and of course going through Information Act and processing a lot of paperwork in order to get this database put together. But he's compiled a great database that allows us to visualize potentially the breadth of the nitrate and uranium problem. And of course, the co-occurrence, it does look like there may be a correlation between the two. So Jason had conducted some additional correlation analyses, geospatial correlation analyses, in order to get a better idea of, is this statistically significant? And what he ended up finding was that yes, there are areas in which there is a statistical significance in which nitrate and uranium are correlated in Nebraska, Wyoming, areas of Kansas, Colorado, and even in Texas, a rather significant correlation has been observed. So there does appear to be this correlation throughout the High Plains aquifer with nitrate and uranium. So let's kind of begin to think about what potentially could this mean. Well, this could be significant in the context if we consider that around a 1 mile radius from these areas in which uranium has been identified, this could potentially influence about 1.3 million people. And so there are a number of individuals that can potentially be affected. So this could be a rather significant problem. So now to understand this correlation between nitrate and uranium and what does it mean, how are the two potentially related? And the first is we have to consider uranium geochemistry. So if there's anything that you want to remember with respect to uranium geochemistry in general, and of course this is a generalized viewpoint of it, is that when uranium is reduced, it's going to exist as an insoluble immobile species, and that's in the tetravalent state. So it's going to be this solid phase uranium mineral. So if you want to think reduced rock, that helps you remember what it is. And that's going to be kind of key because we have to think about uranium in groundwater. In order for it to be removed from the groundwater, it has to be aqueous, and so dissolved in that liquid state. Most often, when uranium is oxidized, it's going to exist as a soluble species. Here I just have it depicted simply as a uranyl carbonate species. So now primarily, and one of the prevailing explanations of uranium in groundwater is that carbonates simply will desorb uranium-6 that had been absorbed to sediment particles, and that's the mobilization mechanism that is employed. The alternative is that uranium-4 minerals are oxidized as a result. So this could be through abiotic processes, and I'm going to talk a little bit today about biological processes that could also promote these reactions. Now, from a perspective of history and of course carbonates and the potential to desorb uranium-6 from the surface of sediment particles, it has been demonstrated that there is this correlation. And that's when I stated that traditionally this is the common thought, is that this is how uranium is mobilized in these environmental systems. However, the oxidation of uranium-4 minerals that might be coupled to the reduction of nitrate or potentially even other oxidants in environmental systems has been identified. And this simply just takes into consideration if we have an oxidant that would result in the oxidation, we're going to promote the formation of an aqueous species. So that could include even a uranyl carbonate species, which would maintain uranium in that mobile state. So just to keep in mind that microbial life is very prevalent beneath our feet. So if you were to walk outside, look down, see the soil, 1/3 of all microbial life, prokaryotic life, is actually in the soils. And then if we think beyond the vadose zone and into the subsurface, there's another third of microbial life. So beneath your feet, that's 2/3 of all prokaryotic life, bacteria, archaea, are going to live in these subsurface environments. And they have to make a living somehow. And of course, they're going to do this by using what energy is available to them. Now, so from a microorganism's perspective, to them, any chemical is essentially food. Most often, we might think of something similar to what we would consume, organic carbon, and of course, breathing oxygen, what we breathe. However, in many environmental systems, oxygen is not available, and organic carbon may or may not be available. So we can simply begin to think about microbial metabolisms, anything that has, in any chemical species, that has a more negative reduction potential can potentially serve as an energy source to any electron acceptor that has a more positive reduction potential. So this is true for microorganisms that could potentially even breathe iron oxide minerals, so they can breathe rust. And inversely, some of those that can eat iron minerals and also generate rust while breathing nitrate or some other terminal electron acceptor as a result. And there's a common concept, even in astrobiology, of follow the energy. So if there's energy out there for an organism to utilize, they're likely going to take advantage of that energy source. And the same is true in the subsurface. So when we actually begin to think about life in the subsurface and how these microorganisms might interact with our contaminant or whatever we're interested in this case, we're going to be talking about uranium biogeochemistry. And there are many organisms, it's been recognized now for over 2 decades, that there are microorganisms that can breathe uranium. And when they do this, they end up precipitating out uranium-4 as a solid phase mineral. And this has actually been used as a biotechnology in order to clean up uranium contamination in mining sites or sites where there is a disposed nuclear fuel that has contaminated the groundwater. And this has been a successful bioremediation strategy. So of course, The inverse is true, and even in natural systems, these bacteria are present and making a living in these environments. But of course, that requires that these environments stay in this reduced, or if we could call it oxygen-free system. Otherwise, that reduced uranium will get reoxidized and again become mobile. Now, of course, there are microorganisms that can play a role in actually mediating uranium oxidation, and it might be indirectly. So you can have a heterotrophic nitrate-reducing bacterium that can produce an intermediate, a reactive intermediate, such as nitrite. And that nitrite could abiotically interact with the reduced uranium, reoxidizing it. Similarly, you could have intermediates of iron oxides that are generated, and this is by bacteria that can utilize reduced iron as the energy source and nitrate as the electron acceptor, and they precipitate out iron oxides, and that iron oxide would then reoxidize that reduced uranium. But also, you can have those bacteria that could use that reduced uranium directly as an electron donor and respiring that nitrate. And so potentially getting energy from this microbial metabolism, mobilizing that reduced uranium. So we're going to talk a little bit about those bacteria that can directly oxidize the reduced uranium. So I've tested two different organisms that I isolated that were originally isolated as nitrate-dependent iron oxidizing organisms. So these are organisms that can oxidize iron as well as reduce nitrate. And I tested them for their ability to also use reduced uranium as an electron donor. I do want to point out that I isolated Acidoborax abreu strain Tipsy from a contaminated site in Oak Ridge, Tennessee. So this is one of their sites at Oak Ridge National Laboratories that is contaminated with spent nuclear fuel. So it has high concentrations of nitrate as well as high concentrations of uranium present at that site. And this organism was isolated from the groundwater. And one of the things that you can see is when you look at this graph, if I give this organism reduced uranium as well as nitrate, you see a significant amount of uranium-6 that is produced as a result. Even with kill cultures, when this is where we kill the cells, we still see some oxidation. So there is an abiotic reaction that also occurs in the presence of nitrate as well. Now, I tested another organism that was isolated from freshwater lake sediments, and this organism is also capable of nitrate-dependent uranium oxidation. Now, one of the things to note is that this red dashed line here depicts the maximum contamination limit. So whether it is the abiotic reaction or it is the biotic reaction, the reduced uranium is substantially oxidized, well above that maximum contamination limit. So biotic or abiotic reactions can play a significant role. And one thing to note too, though, is that the presence of microorganisms, the rate and the extent of the oxidation is much greater in the presence of life than without it. Now, we continued to test a diversity of different organisms for their ability to oxidize reduced uranium coupled to nitrate reduction. So we also identified that other organisms such as Pseudomonas as well as Dichlorospirillum are also capable of oxidizing reduced uranium. Other studies had demonstrated Geobacter metallireducens as capable of oxidizing uranium, and I just want to note that this organism was originally isolated as a metal reducer. So it's commonly attributed that this organism would be a uranium-reducing organism if identified, but it is also capable of nitrate-dependent uranium oxidation. Not only are these organisms diverse as far as their phylogenetic affiliation, but they're also abundant. If we actually look at the number of microorganisms in any environment that might be potentially capable of oxidizing reduced uranium coupled to nitrate reduction, we do find that they are abundant, about 1,000 cells per gram of sediment. But I just want to mention that, and for you to keep in mind, the number here, we're talking about 1,000 to about 3,000 cells per gram of sediment that we identify, whether it is from a Department of Energy contaminated site, noted here by the FRC, so that's Oak Ridge National Laboratories Field Research Center, or if it's freshwater lake sediment or cornfield soil from a cornfield collected here in Nebraska, where we do find abundant um, nitrate-dependent uranium
So
oxidizing
we had conducted
microorganisms.
a study earlier looking
So
at
we had conducted a study
testing our hypothesis that
earlier
both abiotic and
looking
biotic
at
catalysis of
and
nitrate
testing our
and
hypothesis
uranium
that both
for
abiotic and
oxidation
biotic
would result
catalysis
in
of
uranium mobility
nitrate-dependent
and
uranium for
contributing to
oxidation would
uranium
result
in
in
Nebraska
uranium
groundwaters.
mobility and
And I
contributing
conducted this
to
project in
uranium in
collaboration with
Nebraska
Dan Snow,
groundwaters.
and I've had a
And I
variety
conducted
of
this
students,
project in
graduate students,
collaboration
Don
with
Pan,
Dan
Jason Nolan,
Snow,
that have
and I've had a
continued to
variety
work
of
on
different
this
students,
project,
graduate
as
students,
well as
Don
some
Pan, Jason
undergraduates
Nolan, that have
in
continued to
Olivia
work on
Healy
this
as a
project,
bioenergy
as well as
RU student who's now in my
undergraduates,
laboratory as a
and
graduate
Olivia
student.
Healy as a bioenergy RU
We
student
began
who's
this
now in
site
my
at a field
laboratory as a
site
graduate
located in
student.
Alda, Nebraska.
We began this site at a
We
field
had that
site
previously
located in
described
Alda,
as
Nebraska,
having high nitrate and high uranium
because it had been
concentrations.
previously
So our
described
hypothesis was
as
that we
having
might
high
actually be
nitrate and
able to
high
go into
uranium
this environment
concentrations. So
and
our
find
hypothesis was that
reduced
we
or
might
reduced
actually be able to
system
go into this
that would be
environment
present.
and
So the
find
plan was,
reduced
and
a
what we
reduced
did was
system
go out and
that would be present.
collect a core by
So the
geoprobe
plan was, and
coring,
what we did was
geochemically
go out
characterize
and
those sediments
collect a core by
with
geoprobe
four
coring,
valence state of
geochemically
uranium,
characterize those
uranium-4, uranium-6,
sediments
as well as
with
ferrous iron and
for
total
the
iron, as
valent
well as
state of
nitrate.
uranium, uranium-4,
We preserve those
uranium-6, as well
anoxic
as
conditions in the
ferrous
field
iron and
because that
total
becomes
iron, as well
key
as
because we're
nitrate.
interested in
And
the
we preserve those
preservation of that
anoxic
valence
conditions
state.
in the field
In
because
order
that
to
becomes key because we're
know whether
interested
or
in the
not
preservation of
uranium
that valent
is
state
present in
in
that
order
reduced
to
state so we can actually look
know
at
whether or
uranium
not
oxidation.
uranium
We
is
then
present in that
characterize the
reduced state,
microbial
so we can
community
actually
looking at
look
the
at
numbers of
uranium
bacteria that were
oxidation.
present,
We
and
then
that could
characterize
oxidize
the
uranium
microbial
coupled to
community
nitrate
looking at the
reduction, as
numbers of
well as
bacteria that were
then
present
identify the
that could
bacteria that
oxidize
were
uranium
present in
coupled
that
to
system.
nitrate reduction, as well as
So
then
within the
identify the
groundwaters,
bacteria that were present
um,
in that
we
system.
were able to identify that it
So
was a
within the
reduced system.
groundwaters,
The reduction
uh,
potentials below
we were able to
0.0
identify that it
ppm
was a
redox
reduced
power.
system.
This would
The
poise the
reduction
system
potentials
towards an
below
iron producing
zero.
system.
If you look at
So
the
we would
redox
expect
tower,
that we
this
would
would
be
poise the
able to
system
measure
towards an
later
iron
on
reducing
with
system.
iron.
So we would
So of course, when
expect that we would be
being a
able to
reduced
measure
system
later
with
on
reduction potential below
reduced
zero, we wouldn't
iron. So
necessarily
of course,
expect
when,
nitrate to be present.
being a
But
reduced
yet,
system
we
with
do
a
find
reduction
high
potential below
concentrations
zero,
of
we wouldn't
nitrate. We know
necessarily
it's an
expect
anoxic
nitrate to
system,
be present,
but nitrate
but
is
yet we
present
do
across
find
the
high
system.
concentrations
We
of
also
nitrate. So
see
we
that
know it's
there's
an
uranium
anoxic system,
that
but nitrate
typically
is
has
present
been
and
reduced
oxidant is
with
present.
depth.
And we also see aqueous uranium
So this would
that
suggest
has
that the
increased
system is
with
not
depth.
in equilibrium and that
So this
potentially
would
we
suggest
could
this
have
system is not in
nitrate-dependent
equilibrium,
uranium
and that
oxidizing agents
potentially we
present
could
there.
have
When we
nitrate-dependent
looked at
uranium
the
oxidizing conditions
sediment
present.
geochemistry,
When we looked at
specifically
the
looking at iron as well as
the
uranium, what
sediment
we ended up finding is that
geochemistry,
virtually
specifically
all of the
looking at
iron
iron
and
as
all
well
of
as
the
uranium,
uranium
what we
was
ended up
reduced
finding was
in the
that
system.
virtually all of the
So this
iron
was
and all
simply
of the
indicating to
uranium
us that
was
what we're
reduced
measuring, if
in the
we're
system.
going to assume that all
So
of
this was
uranium-6 is
indicating to
indeed
us that what we're
aqueous
measuring, if
uranium
we're
is in
going to assume
the
that all of the
oxidized
uranium
state,
6 is
that
in the,
in the
or the
what
aqueous
we're
uranium
observing
is
in the
in the
sediments, that it's reduced,
oxidized state, that in
that
the, what we're observing in the sediments, that it's
what is
reduced,
happening is nitrate-dependent uranium
that
oxidation. So we do see this
what could
potential
be
for this
happening is
chemical
nitrate
reaction
dependent
occurring in the system.
uranium oxidation.
So
So
we
we
were
do see this
interested if there's a couple
potential
going
for this
on that
coupled
potentially
reaction
there are these
occurring in the
bacteria
system.
that could potentially be
So we were
catalyzing
interested if there's a couple
nitrate-dependent
going
uranium
on that
oxidation.
potentially there are
So
these
what you're
bacteria
looking
that
at
could
is an
potentially be
overlay of all of
catalyzing
the data that we had
nitrate-dependent
collected
uranium
in addition
oxidation.
of
So what
data
you're
from
looking
our
at is an
most
overlay
probable
of all of the
numerations
data
series.
that we
So
had
what
collected
this
with the addition
technique does is
of
allow us to
data
look
from a
at
most probable
bacteria
number
that are
enumeration series.
capable of
So what this technique does
oxidizing
is allow us to
uranium
enumerate
coupled
the
to nitrate
bacteria
reduction.
that are
What
capable
we
of
find is that there
oxidizing the
are
reduced uranium coupled to nitrate reduction.
over
And what we
25,000
find is
cells per
that
gram of
there
sediment
are
located at this
over
site. Now, keep in
25,000
mind
cells
that
per gram of sediment
my previous work
located
had
at this
identified that we
site.
only have at
Now, keep in
most
mind
maybe
that
about
when
2,500 or
my
3,000
previous
cells
work
per
had
gram of
identified that we'd
sediment.
only have
In
at
some of these
most
other
maybe
environments,
about
only about 100
2,500,
cells per gram of
3,000
sediment
cells
in
per gram of
contaminated
sediment in some of
environments.
those
Where
other
here,
environments,
the
and only
graphs
about
of what
100
we're
cells per
observing
gram
is
of
a
sediment in
very
contaminated
microbial
environments, where where here in Nebraska, what we're observing is a very abundant microbial community that is capable of oxidizing uranium coupled to nitrate reduction. So if we actually take a look at the members of this microbial community, what would that tell us? So we conducted some community analyses. And what we ended up observing was that we see an increase in members of the Comenidaceae as we go with depth. Now, members of the Comenidaceae, specifically an acidivorax species was identified. Now, what does that mean? Well, I had previously isolated an acidivorax as a metal oxidizing microorganism, and this is the one I talked about, and introduced it as a nitrate-dependent uranium oxidizing microorganism. So this indicates that there are microorganisms that are present and actually make up about 50% of the microbial community that is present in this environment. And so we know that potentially these organisms can be playing this role in oxidizing uranium in this reduced environment. So we know the conditions are present, and we know that there are microbiota that are abundant, as well as members of microbial communities that are physiologically capable of oxidizing the reduced uranium. So we've begun to put together a conceptual model as we begin to think about how uranium could be mobilized in these Nebraska groundwaters. However, one of the things, I don't really have time, unfortunately, to go into this, is that we do see a lot of uranium reduction also going on in this system. So it's not simply just a story about oxidation, but it's also a story about reduction and uranium reduction occurring within these sediments. So uranium can get reoxidized, but it's also going to get re-reduced as well. So there's uranium biogeochemical cycling that's ongoing in the system. So I think that what's actually going on in this system is that it's complicated by the kinetics of these reactions and how groundwater flow rate and withdrawals might potentially also be competing with the kinetics of these overall reactions in these subsurface systems. So there's a lot more to study that we don't know as of yet. And in the end, we still are left asking, what does this mean for uranium mobility in Nebraska and other regions of the United States, as well as thinking about the city of Hastings? And ideally, we need to know, where is that uranium coming from? Is it in the sediments? We focused on the sediments. However, within the surface soils, is there the same or similar biogeochemical processes that would be going on in the soils, and there's their uranium transport from the vadose zone down into the subsurface? And in the end, if uranium is existing even in the surface soils as a reduced state, we don't want oxidation, solubilization of that uranium, and want to prevent this. So we need to understand a little bit more, and hopefully, as we move forward in collaboration with Marty Stange, that we get a better idea of where is the uranium coming from, and potentially how can we actually have a better understanding of the kinetics of this system and controls over uranium mobility when we take into consideration the biogeochemistry and these natural processes that are going to be happening. Because after all, if we could let nature take its course so that the bacteria that are present would naturally keep the uranium reduced and insoluble in the subsurface environment. So there have been a variety of individuals that have collaborated on this project. So with that, I thank everybody. Members of my laboratory. Jason Nolan's a current PhD student working on this project. Collaborators, Dan Snow, my former collaborator at the University of California at Berkeley, and Marty Stange, who we're beginning a new collaboration with to understand uranium mobility.